Hello, everyone. Here is episode number 2, Primary Separation Modes of HPLC. Here is the agenda where we describe the 4 primary modes of HPLC, their mechanisms and applications. First is normal phase chromatography or NPC. Second is reverse phase chromatography or RPC. It is highlighted because it's the most common mode. Third is ion exchange chromatography, IEX. Finally, size-exclusion chromatography, or SEC. In this slide, we describe the chromatographic process where the sample mixture is separated into its constituent component by distribution between the mobile phase and the stationary phase. Diagram A shows the general overall chromatographic process where we have a column packed with solid support and a mixture of sample which contain two components A plus B are being eluted with the mobile phase traveling down the column. Component B is much more retained by the stationary phase, therefore is eluding later and being detected as a uh, later eluding peak. In diagram B, we show you a microscopic view of the analyte retention. Shown here is a single solid support stationary phase, typically a porous silica material, bonded with some type of stationary phase. One can see the analyte A and B represented as single molecules of the analyte traveling in the mobile phase of flow rate. And B is much more retained because it is have higher partitioning coefficient or retention mechanism into the solid support. Therefore, in the chromatogram, where we have absorbance versus the time, component B is eluding as a later eluding peak. In this slide, we demonstrate the schematic retention mechanism of mode number 1, normal phase chromatography, generally called absorption chromatography. In the left-hand side, we see a silica particle, which is the most common support for normal phase chromatography. And the stationary phase are actually the residual cyanide groups residing on the surface of the silica support. These could be on the surface or could be inside the pore of the particle. In normal phase, the mode is called absorption or polar interaction. Here, a mobile phase generally being used is hexane modified with different amount of alcohol. Polar analyte in the sample is being retained more because of a polar interaction of absorption by the residual cyanide groups. So in normal phase chromatography, the most common station phase is silica, or actually the residual cyanide group on silica, using hexane modified by alcohol as the mobile phase. The common application are separation of isomers and for purification of organic materials. However, there's a very bad disadvantage because they're not used in quantitative analysis due to poor reproducibility due to column contaminations. In this diagram, we show you a illustration case study on the separation of isomers of vitamin E using normal phase chromatography. Detection is using fluorescence detector with a sample of crude palm oil, and we saw 4 components of the palm oil sample being separated in about 5 minutes. Here's the operating condition. A column being used is the silica column with the dimension shown here. Mobile phase is hexanes modified with a little bit of ethyl alcohol flow rate. detection by fluorescence. Sample is 5 microliter or 0.1% of a crude palm oleins dissolved in hexanes. Here's the 4 different separated components, starting with vitamin E or alpha tocopherol, followed by 3 isomers of tocotrienols, meaning that with 3 double bonds in the molecules. Normal phase chromatography is less reproducible due to column contaminations from absorption of some ley eluded on top of the column. This issue can be somewhat mitigated, but not completely eliminated by the use of polar bonded phases. Normal phase, however, is important in the separation of isomers by supercritical fluid chromatography and also in chiral separations. Mode number 2 is reverse phase chromatography, or RPC, also known as partition chromatography. Shown in the left-hand side is a silica particle with pores inside and bonded with a C18 hydrophobic ligand, shown here, octadexyl silane ligand, shown here. So the surface of the station phase is definitely hydrophobic or non-polar. We are always using a mobile phase consisting of aqueous mobile phase, either buffer or acidify or basify. And in this case, the non-polar analytes would be retained by higher partitioning coefficient into the hydrophobic stationary phase. In reverse phase chromatography, is the separation mode is partitioning or hydrophobic interaction. Perhaps solvophobic interaction is a better term, since the analyte actually is interacting with any mobile phase molecules in the mobile phase, such as water by hydrogen bonding, acetonitrile or methanol through different type of solvent and analyte interaction. The most common station phase in reverse phase is the C18 bonded phase, bonded to porous silica support. And the mobile phase used is a mixture of water with methanol or acetonitrile. Reverse phase is used in 80% of all the HPLC application. The retention mechanism is hydrophobic interaction, or the partitioning of the analyte into the hydrophobic stationary phase. Perhaps the term solvophobic interaction is a better term, since the mobile phase components play a role in the retention mechanism of the analytes. Here is an application on reverse phase chromatography, shown here in a separation of a mixture with 3 components. A first component is uracil, which is something called a void marker, designating the void volume or the void of the column. The second one is pyridine, which is a basic component, which generally shows some type of peak tailing due to interaction with silica surface. And finally, more hydrophobic tertiary butylbenzene, even though that the peak is broader, the peak is symmetrical for most of the time for neutral analytes. The operating conditions are shown here with a column being a C18 column with 32 millimeter length, 4.6 millimeter ID. Mobile phase, a mixture of methanol and water. The flow rate, the pressure, the detection wavelength, and the sample, which is a mixture of 3 different organic components. Reverse phase is the dominant mode in pharmaceutical analysis and is highly reproducible. Very important to know that peak do broaden with retention time under isocratic condition. Is called column band broadening, which is fundamental in chromatography. Basic analyte often tail due to interaction with silica support. This slide summarizes why reverse phase is the most common mode and why do we focus on reverse phase chromatography in HPLC training. So number one is used in 80% of all LC application, particularly dominant in pharmaceutical analysis and bioscience research because it's a good fit with the analyte type. Also, reverse phase can be used for mass spectrometry when volatile additives in the mobile phase are used. And in this case, it is called mass spec compatible operating procedure or analytical methods. Number four, reverse phase have excellent reproducibility and also very good mass balance for purity analysis. Therefore, is used in pharmaceutical analysis, particularly to monitor the purity of drug substance or drug products. Here we showed mode number 3, or ion exchange chromatography. In the right-hand side, we showed a particle of ion exchanger based on either polymeric, or silica support. In this case, it is a cationic exchange where we bond a negative anion ligand, in this case sulfate, to the surface of the polymer. And analyte in the sample, exemplified by these P plus cation, because any cationic component in the sample would exchange or interact with the sulfate anion and become a pair ions. And these are being pushed out during the chromatographic process via the mobile phase pH and also the mobile phase salt content. So in ion exchange mode, the retention mode is ion exchange or ionic interaction. Okay, in IEC, the mode of retention is ionic interaction or ion exchange of the ionic analyte with the bonded charge ligands in the stationary phase. For instance, in cationic exchange shown here, the bonded negative sulfate ligand retain any cationic analyte which are eluted with a mobile phase of increasing concentration of sodium chloride to kind of displace the bounded cation analyte ions, or operating at higher pH where you lose the charge of the if it's a weakly positive ion, weakly basic ion, it would lose its charge. Major application of ion exchange chromatography are ion chromatography, IC, and also the separation of proteins and amino acids. In this chromatogram, in this diagram, we show an application of ion exchange chromatography of primary amino acids, or AA. Now, since amino acids are not retained by reverse phase, ion exchange is a better mode for retention. And also, since amino acids are not chromophoric, does not absorb UV, we need to use something called a post-column derivatization technique, typically with anhydride, in order to get absorbance in the visible wavelength. Here is the chromatogram of 20 essential amino acids separating. The concept of the ion exchange chromatography actually was demonstrated in the 1990s by two Nobel laureates, which are biochemists in New York City, Rockefeller University, published in Analytical Chemistry. This chromatogram is a modernized version shown by Pickering Laboratory using different reagents or buffers, different columns, and also different derivatization reagent. Here are the column and operating detail. Sodium cation exchange column, mobile phase using 3 different buffer of increasing pH, increasingly higher pH, the flow rate, the column temperature, and also detection using post-column reaction with a reagent called triome, which is similar to anhydrine, except it's more stable. In the mobile phase, we use different increasing concentration of salt, sodium chloride, and also increasing pH to displace the bounded amino acid ions. Ion exchange chromatography was used in the first developed amino acid in the 1950s, which actually predates or is the predecessor of modern HPLC. Here we come to mode number 4, which is size-exclusion chromatography, or SEC. Shown here on the left-hand side is a particle of control pore support material. Here, there's no interaction of the stationary phase with the analyte, with the exception of a process called size-exclusion, where the larger molecule are eluted first because they cannot penetrate the smaller channels, the smaller pores, while these smaller analytes in the sample actually would elute later because it actually can penetrate all the pore in the support material. So the mode is called size exclusion uh, and separation by molecular size, since the larger molecule cannot enter the smaller pore of the packings and are eluted first. The major application are the separation of organic polymer for it's called GPC, and also separation of biomolecule by size, such as proteins. Here's an application of size exclusion chromatography of organic polymers such as polystyrenes. And here is the GPC chromatogram showing that the less component, which is the diluent of the sample, actually eluded last, actually eluded at the solvent front. Everything eluded before the solvent front. And therefore, the resolution is actually not very good because you don't have a lot of retention volume in the chromatography. Here's the detail. A column is using a gel with this dimension. Mobile phase is THF, a flow rate and detection by UV at 265. Sample ID containing these components, starting with the first one, which is a high molecular weight polystyrene, with some of the polystyrene standard or all the way down to an oligomer with molecular weight of 550s. And number 9, ochlorobenzene, is the diluent. So in size exclusion, the large molecular weight component eluded first due to exclusion from smaller pores. Size exclusion has lower resolution than reverse phase. It is called GPC, or gel permeation chromatography, for the use of organic polymer separations. Here's my summary and conclusion. The 4 primary modes of HPLC and application are normal phase chromatography or absorption chromatography for the separation of isomer and purification, reverse phase or partition chromatography with hydrophobic interaction as a retention mechanism is the most common mode used in most applications, including most pharmaceutical analysis. Ion exchange using ion exchange resins, either in the cationic or anionic mode, with mobile phase of increasing salt strength and also different pH to elute the ionic analytes. Ion exchange chromatography is used in the separation of ions in ion chromatography or separation of protein and amino acid. Also used in quality control of biopharmaceuticals. Finally, a size exclusion chromatography using a control pore support to separate large molecule based on molecular size. SEC used in separation of organic polymers or biomolecules. Here is the references and also links for more information on the various websites and subscription to the YouTube channels. Finally, there are some quizzes with actually the answers being marked here to test your retention of these concepts. On that note, thank you for your attention and see you next time.